Hello, my dear pharmacy students. My name is Sachin Jadhav, founder Fundamental Pharmacy, author of Fundamentals of GPAT Niper DI Pharmacist Volume 1, GPAT Cracker Achiever MCQ book, and few other books. Welcomes you in this YouTube video session. My dear students, if you are new on our channel, please subscribe the channel and press the bell icon. In this video session, we are going to see important pinpoints from pharmacognosy, which are very important for GPAT, Niper, Drug Inspector and Government Pharmacist exams. We have already completed 3 videos on pinpoints of pharmacognosy. If you have not seen those videos, you can go and see those videos also. The link of that videos also will be available in the description box of the. In the last video, we have seen the introduction classification of glycosides. So in today's session, we will see the important chemical test, important identification test for glycosides. We have seen the different classes like Anthrochromium glycosides, saponin glycosides, cardiac glycosides, flavonoid type of glycoside, steroidal glycoside, triterpenoid glycosides, coumarin glycosides. So these are the different classes of glycosides. Now, what are the important identification tests for these glycosides? Because these identification tests they have already asked in a lot of times in the various exam. So you can expect either a direct question on the identification test, or you can expect match the following type of question. Now first we'll see identification test for anthraquinone glycoside. So what do you mean by anthraquinone glycosides? So anthraquinone glycosides will have anthraquinone in the structure, anthracene ring plus 2 ketone group. So that is the anthraquinone glycosides. Those glycosides which will have anthraquinone structure in the ring in their molecule, they are known as the anthraquinone glycosides. So now there are 2 tests for anthraquinone glycoside. So first one is the Braun-Trager test and second one is a modified Braun-Trager test. So remember over here, Braun-Trager test is a specific test for O type of glycosides. Specific test for O type of glycoside. And modified Braun-Trager test is a specific test for C type of glycoside. Now in case of Braun-Trager test, what happens? You have to take a powder drug and add dilute H2SO4 in the test tube. Then filter it, and to a filtrate you have to add organic solvent like benzene or ether or chloroform. Then you have to shake well that test tube. Now you have to separate the organic layer by using separating funnel. Separate the organic layer and add ammonia to the organic layer. Now ammonical layer will show pink color. Remember pink color, that is due to the presence of anthraquinone glycoside. And remember, it is a specific test for O glycosides. That means anthraquinone glycosides where there is a oxygen is involved in the glycosidic bond. So O type of glycosides will be give this test positive. And modified Braun-Trager test, in this case you have to take powder drug plus add hydrochloric acid, then add 5% ferric chloride, boil it for 5 minutes, then extract with the carbon tetrachloride or benzene, and then separate, and you have to add ammonia. So ammonical layer will show pink color, pink or rose red color, and due to presence of anthraquinone moiety. Modified Braun-Trager test is for specific for C type of glycoside. Remember. Now hemolytic test and foam test. So these 2 tests are for your saponin type of glycosides. So hemolytic test and foam test. Hemolytic means hemo means blood and lytic means breakdown. So this test involves the observation of breakdown of blood. Okay, RBC. Few drop of blood on slide, mix it with a few drops of saponin solution. RBC will become ruptured. That can be observed in the microscope. So that is the hemolytic test. Then foam test, a 1 gram sample of glycoside and add 10 to 20 ml water in the test tube, and for few minutes you have to shake it. Now formation of foam or frothing which remains for around 60 to 120 seconds, which will indicate the presence of saponin. So remember, Braun-Trager test is for O glycoside, modified Braun-Trager test is for C type of glycoside, foam test and hemolytic test is for saponin glycosides. Okay, so anthraquinone glycosides we have seen, saponin. Now next we will see the steroidal glycosides or triterpenoid glycoside. Identification test for steroidal type of glycosides or triterpenoid glycosides. So in this glycoside, steroid ring will be there or triterpenoid ring will be there. Now for that you have this test. First one is known as a Salkowski test, then Lieberman-Burger test, Third one is the antimony trichloride test. Fourth one is a trichloroacetic acid test. Fifth one is a tetranitromethane test, and fifth one is a Zimmermann test. So all these tests are the tests for identification of steroid nucleus or triterpenoid glycoside. Steroidal or triterpenoid glycoside. Remember the name of test. First one is a Salkowski test. Second one is a Lieberman-Burchard test. Third one is a antimony trichloride test. Fourth one is a trichloroacetic acid test. Fifth one is a tetranitromethane test, and fifth one is a Zimmermann test. Now, in case of Salkowski test, you have to take alcoholic extract of drug and you have to evaporate that extract to dryness. Then you have to add chloroform. Then you have to add chloroform and extract. Then you have to add concentrated sulfuric acid from the side wall of test tube. So formation of yellow color at the ring of junction at the junction of two liquids, which will turn into red. That yellow color will turn into red after few minutes. That will indicate the presence of steroid nucleus. Okay, this is the test for identification of steroidal type of glycoside. Now, second one is known as a Lieberman-Burchard test. So again, in this test, you have to take alcoholic extract of drug, then evaporate to dryness and extract again it with the chloroform. Then add concentrated sulfuric acid from the side wall of the test tube. Again, you will get violet blue color ring at the junction of two liquids. That will indicate the presence of again steroid nucleus. Okay, so Salkowski test, Lieberman-Burchard test for the steroid nucleus. Okay, remember. Third one is the antimony trichloride test. So again, alcoholic extract you have to make of your drug, evaporate to dryness, and again extract with the chloroform and add saturated solution of antimony trichloride in a chloroform containing 20% acetic anhydride. Now, formation of pink color on heating indicate the presence of steroid or triterpenoid. Okay, that is the antimony trichloride test. Next one is a trichloroacetic acid test. So it depends on the addition of saturated solution of trichloroacetic acid and forms colored precipitate. Means you have to take a solution of your crude drug and add trichloroacetic acid, saturated solution of trichloroacetic acid. It will form a colored precipitate that indicate the presence of steroid nucleus. Now, tetranitromethane test. So with the steroid glycoside, you will get a yellow colored with which is generated. Yellow color is generated with the steroid and triterpenoid glycosides. So addition of tetranitromethane with the drug, you will get a yellow color. Now next one is a Zimmermann's test. So in this test, meta-dinitrobenzene solution is added to a alcoholic solution of drug containing alkali. It will form a violet color in presence of particularly keto type of steroid. So these are the 5 tests for steroidal type of glycoside: Salkowski test, Lieberman-Burchard test, antimony trichloride test, trichloroacetic acid test, tetranitromethane test, and Zimmermann test. Now we will see test for cardiac glycosides. So इसके ऊपर भी already question आ चुके हैं. So what are the different tests for identification of cardiac glycoside? So first test is the Killer-Kilani test or Keller-Kilani test for digitoxos. It is a specific test for specific sugar found in the cardiac glycoside, that is a digitoxos. So Keller-Kilani test for digitoxos. Then legal test is there. Then Baljet test is there, and 3,5-dinitrobenzoic acid test is also there. So these are the 3 tests for cardiac glycosides. Now first we'll see Keller-Kilani test. So in that you have to take 1 gram of powder in test tube and add 10 ml of 70% alcohol. Then boil the solution for 2-3 minutes and filter it. Then add 5 ml of water and 0.5 ml of strong lead acid solution and shake the solution. Filter it, and to a filtrate you have to add equal volume of chloroform and evaporate it. Now extract it. Now evaporated extract is dissolved in the glacial acetic acid. Okay, and then you have to cool the solution and add a 2 drop of ferric chloride and transfer this solution into another test tube containing 2 ml of sulfuric acid. You will get a reddish brown layer. Acquiring bluish green color after standing. and that is due to the presence of digitoxos. So remember, Keller-Killarney test is specific test for digitoxos. Now second test for cardiac glycoside, legal test. Legal test, in that test you have to make an extract of drug and add pyridine to extract and plus sodium nitroprusside solution. Shake, you will get a pink or red color, and that indicates the presence of cardiac glycoside. Now third test is nothing but the Baljet test. Third one is a Baljet test. Now in case of Baljet test, you have to take a section of digitally sleep, add sodium picrate, you will get a yellow to orange color. That is a Baljet test. Now fourth test is nothing but the 3,5-dinitrobenzoic acid test. So alcoholic solution of your drug plus few drop, you have to add few drop of sodium hydroxide followed by 2% solution of 3,5-dinitrobenzoic acid. You will get a pink color, and that is due to the presence of cardiac glycoside. So cardiac glycosides, they are nothing but the cardiotonic. They are cardiotonic, digitalis, Tibetia, okay, such kind of glycosides. So they will give you positive these tests. Keller-Killarney test, specific test for digitoxos, then legal test, then Baljet test, and dinitrobenzoic acid test. Now we'll see the test for coumarin glycosides. Now test for coumarin glycosides. So what are the tests for coumarin glycosides? So first one is a ferric chloride test, and second one is a fluorescence test. So in the ferric chloride test, concentrate alcoholic extract of drug plus few, you have to add few drop of ferric chloride solution. Formation of deep green color which will turn into yellow on addition of nitric, okay, nitric oxide HNO3. That will indicate the presence of coumarin ring. So this coumarin glycosides will have coumarin ring, and that coumarin ring will give you deep green color which will turn into yellow with this ferric chloride test. Fluorescence test. So alcoholic extract of drug, you have to mix it with the 1 normal sodium hydroxide. You will get a green blue color which is the result of presence of coumarin glycosides. Okay. For next glycoside, next one is the flavonoid glycosides. Now we'll see the test for flavonoid glycosides. So there are three tests. First one is the ammonia test. Second one is the Shinoda test. Third one is the vanillin hydrochloric acid test. So already they asked the question on this Shinoda test. Okay, for flavonoid glycoside. Ammonia test. First, filter paper you have to dip in the alcoholic solution of, and it is exposed to ammonia vapor. So alcoholic solution of drug, you have to make alcoholic solution of drug, and you have to insert or you have to weight the paper using that solution, and now that paper is exposed to a ammonia vapors, you will get a yellow spot on the filter paper. That is known as the ammonia test, and specific for flavonoid type of glycosides. Now Shinoda test, they have already asked the question on this. Alcoholic extract of drug, you have to add magnesium, then add dilute hydrochloric acid. So you will get a pink color, pink or red color formation. That indicate the presence of flavonoid glycosides. Again, there is a two methods. Second method is alcoholic extract of drug. You have to add zinc turning, then add HCl. You will get a deep red color due to flavonoid glycoside, dihydroflavonoid. So remember, Shinoda test is a specific test for flavonoid glycosides. Next one is a vanillin hydrochloric acid test. So alcoholic solution of drug plus vanillin hydrochloride, you will get a pink color due to presence of flavonoid. So remember, for flavonoid, ammonia, Shinoda test, and vanillin hydrochloric acid test. For coumarin glycosides, you have ferric chloride and fluorescence test. For cardiac glycosides, you have Keller-Killarney test, legal test, Baljet test, and dinitrobenzoic acid test. So ये Keller-Killarney test के ऊपर भी क्वेश्चन आ चुका है, Shinoda test के ऊपर भी क्वेश्चन आ चुका है. If you want such kind of lectures, full lectures, so you can join our GPAT Niper 2023 online course. Here you will get live lectures on Wednesday, Saturday, Sunday. Recorded lectures will be also available in the application. Study material will be given to you in the form of hard copy. Online test is also available. All this included in the 8,000 rupees. We have very much promising result in the past previous years. So thank you very much for watching this video.